Hi friends, today we are going to see the past year NEET questions for the chapter Biomolecules, and in this video we will cover part 2. Now let's begin with the first question here. This question, we are beginning with the 2000 question. Okay. Conjugated proteins containing carbohydrates as prosthetic group are known as dash. So before we go into the solution, we should know what is conjugated proteins. How can you describe them? A conjugated protein is a protein that functions in interaction with other chemical groups, and they are attached by covalent bonds, or when you say covalent, it is sharing of electrons. So the interaction is weak. Many proteins contain only amino acids and no other chemical groups, and they can be called as simple proteins. Okay. Now conjugated proteins containing carbohydrates. So it's very clear. Out of the 4 options given here, chromoproteins depends upon the color. Lipoproteins are the proteins associated with lipids. Nucleoproteins are associated with nucleic acids. And glycoproteins is the only protein which is associated with carbohydrate. Therefore, the correct answer to this question is option B, glycoproteins. Okay. So that is what they have explained in the solution part. Nucleoproteins are associated with nucleic acids. Chroma means they are attained with pigments. Okay, therefore the correct answer is option B, glycoproteins. Next question here. Which is an essential amino acid? Again, it's a 2000 question. Essential amino acids. Now, what is amino acid? You should know that, right? Now, amino acid is a simpler form. When protein is getting converted into a simpler form, it will get converted in the form of amino acid, right? So that is what we are talking here. Which is an essential amino acid? Essential amino acids are those which cannot be synthesized in the body. So when you say essential, it has to be provided by the food or the diet that we take. Okay, it has to be provided from the outside source. It will not be synthesized by the body. So essential amino acids are those which cannot be synthesized in the body from precursors. We do not have any precursors which will do that activity. Okay, so these kind of amino acids are called as essential amino acids. When we say non-essential, it is understood, then they will be synthesized from the body. Therefore, the correct answer to this question is phenylalanine. While if you take serine, glycine, aspartic acid, all these are non-essential amino acids which are present in the body itself. They can be synthesized from the body itself. Okay, so the correct answer to this question is phenylalanine. They are valine, leucine, isoleucine, lysine, then you have methionine, phenylalanine, tryptophan. All these are called as essential amino acids. They have to be produced from the outside source. It can be in the form of any food or diet. Okay, now here if you see, you have a picture which shows the amino acid functions. What are the functions of amino acids given here? It is extrastructural function which helps in collagen, movement function, immune function, hormonal function, digestive function, nutrients transport. So proteins are essential, or amino acids are essential for all these actions. Okay, the correct answer is option D. Now let's move to the next question here. This question was asked in the year 1999. The secondary structure of that portion of an integral protein that is buried in the lipid bilayer of a cell membrane is dash. Okay. What do you mean by secondary structure? Right? What do you mean by secondary structure? They're asking us the secondary structure of that portion of the integral protein. Where it is buried in the lipid bilayer. Where is the lipid bilayer? Phospholipid bilayer, all that is in the endoplasmic reticulum. Secondary structure is the development of the new steric relationship amongst the amino acids through the formation of intra and interpolypeptide hydrogen bonds. Okay, you're given 4 options here: beta strand, beta bend, random coil, and alpha helix. The correct answer goes to alpha helix. If you want to know something more about this alpha helix, it is just a shape, right? It's just a shape of how it is helical, all that we tell, right? The alpha helix is a common motif in the secondary structure of proteins, and it is called as right hand helix. I think all of you knows this. Right hand, we have the right hand and the left hand thumb rule, right? Likewise, this is a right hand helix. Okay, then you have a diagram that I've told you, which I've given you, which is actually representing alpha helix. So it's a right hand principle rule which follows here. It has 3.6 residues per turn. How many residues per turn? 3.6 residues per turn. Each amino acid will rotate at a helix of 100 degrees, and typical length of each typical length of a alpha helix will be, or it will consist of, it will consist of 10 amino acids. Okay, so these are just extra information that you need to know. It might help you, right? So the correct answer is option D. Next question here. This question asked in the year 1998. The enormous diversity of protein molecules is due mainly to the diversity of dash. Okay, this is an easy question for you. Okay, they're asking us about the protein molecules here, and they're asking us on what basis the diversity is dependent on, right? The enormous diversity of protein molecules is due mainly to the diversity of amino acid sequence within the protein molecules. I just now told you what is amino acid, right? The conversion of a protein to a simpler form is called an amino acid, and you have 20 essential, there are about more than 20 amino acids, but our body has about 20 amino acids, and these can be arranged in different order in the polypeptide chain, which form a diverse array of proteins. Therefore, the correct answer to this question is amino acid sequences within the protein molecule. You always have to remember they are talking in group. They are not talking about a single amino acid where you have to talk about peptide bond, but they are asking in general the protein molecules. Then you have to take the amino acid sequences within the protein molecule. The correct answer is option C. Next question here. This question was asked in the year 1997. Genes are packed into a bacterial chromosome with the help of what? This is another easy question for you. If you know about bacteria, this will be easy for you to answer. So what is bacteria? It is a microorganism. We all know that. Micro means what? Something that cannot be viewed under your naked eyes. You have to use a special system or a special machine called microscope, which will help you clearly make it visible to your eyes so that you can see it. Now, genes are packed into the bacterial chromosome with dash. In bacteria, the chromosome or the nucleoid is naked. They don't have a proper cell wall. We all know that. And it has a double-stranded DNA also. The bacterial DNA is folded with the help of RNAs, forming a supercoiled circular complex. The coiling is stabilized with the help of a non-histonic proteins. So it does not have any histones. We have histones, right? Eukaryotics have histones. But here it is a non-histonic protein. So it should be basic proteins. Actin, it is associated with microfilaments, and acidic proteins cannot do that activity. So the correct answer is basic proteins. See, when it comes to bacteria, always remember it is non-histonic proteins. Non-histonic is the other name for basic proteins. Okay, the correct answer is option B, basic proteins. Okay, next question here. Protein synthesis in an animal takes place through dash. Okay, so we all know that when we talk about the endoplasmic reticulum, it has two parts, which is the rough and the smooth endoplasmic reticulum. When we talk about the rough endoplasmic reticulum, it synthesizes proteins, and when we talk about smooth endoplasmic reticulum, it synthesizes lipids. Now we are talking about the protein synthesis in an animal takes place in dash. In cytoplasm, protein synthesis is restricted to ribosomes. Mitochondria is a site of ATP synthesis, and it is also called as a semi-autonomous organelle. Hence, what do you think will be the formation now? We are talking in case of animals. We are not talking in case of humans here. And here, if you see, it will have, when I say semi-autonomous, it is partly dependent and partly independent. That is the meaning of that. And it has its own genetic machinery to synthesize proteins. Therefore, it is in association with two here. When we talk about eukaryotes, always remember that when we talk in endoplasmic reticulum synthesis, that is different. But here they are asking the protein synthesis in animal cell takes place in dash. It is in the cytoplasm as well as in the mitochondria. The correct answer to this question is option C. See, in cytoplasm, protein synthesis is restricted to ribosomes, and mitochondria being semi-autonomous organelle, it can help in synthesis of protein also. Therefore, the correct answer is cytoplasm as well as mitochondria. Next question here. This question was asked in the year 1992. Amino acids are produced from dash. Okay. So where do you think amino acids will be produced from? Now I told you what is amino acid. It is a simpler form of a protein, right? We also have amino acids separately for the lab purposes, for research purposes and all that. Apart from that, what you can describe about amino acid, it is an organic compound that will contain an amine and a carboxyl function group along with a side chain which is specific to each amino acid. As we already know, our body itself has more than 20 amino acids which help in a lot of other functions. Now what all it will contain, if you see, it will contain a, what does it contain? It contains a amine group. What else it contains? It contains a carboxyl group. Okay, then it contains a side chain along with a side chain which is specific to each amino acid. The key element of an amino acid is, it is carbon, hydrogen, oxygen, nitrogen, although some elements which are present in trace amounts. We already saw in the earlier part where we spoke about the 6 essential elements for a living organism in together, where we saw potassium and calcium also plays a major role in that 6 element category. But when we talk about this amino acid sequence, or particularly amino acid, these 4 things are very essential for it: carbon, hydrogen, oxygen and nitrogen. So this is just a basic introduction about amino acid that I'm giving you. So where is it produced now? See, I told you, proteins are converted into a simpler form of amino acids. Therefore, proteins cannot be the answer. Fatty acids, it's very well understood, it is of lipids, so it cannot be the answer. Essential oils, again, it's a lipid, it cannot be the answer. Therefore, the correct answer is alpha keto acids. An amino acid molecule consists of a central carbon bounded to a carbon group and a carboxyl group. That's what I said, amine and a carboxyl group. And therefore it is produced from the alpha keto acids. The correct answer is option D. Okay, next question here, which was asked in the year 2016. A typical fat molecule is made up of dash. So what is the question here? A typical fat molecule is made up of dash. Fine. Here I've given you I've given you a clear-cut information, and apart from that you also have a figure that will represent that. Fat molecules are made up of atoms which comprise of carbon, hydrogen and oxygen. In the previous question we saw that an amino acid requires carbon, hydrogen, oxygen and nitrogen. Now this requires 3 elements mainly. The hydrogen and oxygen atom forms, the atoms will bind to the carbon as pictorially depicted. So you can see how it is bonded here. Can you see it here? So this is how it will form. It is chain of carbon and hydrogen. So you can see here. So a typical fat molecule will have 1 glycerol and 3 fatty acid molecule. So what does a typical fat molecule made up of? 1 glycerol and 3 fatty acid molecule. The correct answer is option B is the right answer. So you have the structure which depicts here a chain of carbon, hydrogen which is there, and you also have a glycerol to your left hand side. You have a glycerol, then you have 3 fatty acids which are attached to this. You can see it here. So the correct answer is option B. Next question here. The question is small but the options are quite big here. So this question was asked in the year 2013. A phosphoglycerate is always made up of dash. What is it? A phosphoglycerate is always made up of dash. So here if you see, what is phosphoglycerate, first of all? You should know that, right? Phospho means it is attached with phosphorus group. Now how about glycerol? Glycerate. Phosphoglycerate. Now phosphoglycerate can be described as having a phosphate group at the third position. So which position? The third position. Okay, and remember this, it is an intermediate in the metabolic pathways like glycolysis and Calvin cycle. It has roles as a fundamental metabolite and also plays an important role in algae metabolism as well. So see how important it is. Okay, now coming to the question here, a phosphoglycerate is always made up of dash. You're given 4 options here. Okay, a saturated or unsaturated fatty acid esterified to a glycerol molecule to which a phosphate group is also attached forms phosphoglycerides. Phosphoglycerides are the major structural component of cell membranes and therefore they are called as membrane lipids. Okay, so here the correct answer goes to which one? Option B. A saturated and unsaturated fatty acid when they are esterified to a glycerol molecule to which a phosphate group is also attached to it. Therefore the correct answer will be option B. Okay, the rest will tell you that it is only one unsaturated acid or a saturated or an unsaturated fatty acid. Therefore the correct answer you have to remember here is it is a saturated or unsaturated fatty acid. Okay, next question. Now this question was asked in the year 2002. Lipids are insoluble in water because lipid molecules are dash. This is very easy question for you because it is hydrophobic, right? Lipids do not dissolve in water. They are hydrophobic. They are water hating. Okay. Water attracting molecules are called as hydrophilic. We all know that. We will call that sequence as water loving. Right? We will call it as water loving. Next, water attracting molecules are called hydrophilic, water repelling molecules are called as hydrophobic. Amino acids simultaneously carry a positive and negative charge. Such substances are called as zwitterions. They contain both the charges, right? You will call them as zwitter, which has 2 charges in it. Lipids are compounds which have carbon, hydrogen and oxygen, but the ratio of hydrogen and oxygen is more than 1. That is, it will be 2:1. So what happens here is lipids become insoluble in water but soluble in a non-polar solvent such as benzene, chloroform, etc. Therefore, this is an easy question for you to understand, right? Lipids are insoluble in water because they are water hating. They are hydrophobic. The correct answer is option B. Okay, next question. This question was asked in the year 2001. Spoilage of oil can be detected by which fatty acid? Okay, this is a question that you need which is essential for your life as well. Sometimes we always think oil doesn't get spoiled. We always happen to have that in the mind because oil will not get spoiled, but it gets oxidized in the air. That you can remember. See, imagine oils are always kept in airtight containers so that they don't have much contact with air. Imagine the oil has more contact towards air, it will get oxidized. That is why once you cut open the oil from a packet or a bottle, you cut open the bottle, when it starts getting exposed to the air, automatically the oil will not sustain its life for a long time. You will get a smell which will clearly tell you the oil is not in a good condition to use. Now the spoilage of oil can be detected by which acid? If you see, it is rusik acid. Rusik acid, which is option D. Rusik acid is a monosaturated omega-9 fatty acid which is denoted by 22:1. Okay, it is actually prevalent in rapeseed. Then you have in other seeds as well. Mustard seed also has this acid, making up to 40 to 50% of their oils. Now this rusik acid is also known as cis-13. Okay, cis-13. So here it is. So you can clearly see it here. Rusik acid is also known as cis-13 docosanoic. Okay, docosanoic acid, and its trans isomer is known as brassidic acid. So here you need to know is, see, This rusik acid is already present in oil. So when the content seems to increase, you can identify the oil is spoiled. So this is one acid which will help you detect the spoilage of oil in the fatty acid. Therefore, the correct answer to this question is rusik acid. I want to tell you an example here. Sometimes we all have the tendency to buy potato chips, or basically chips, from the market, and you always see them. Their covers will be air filled, and when you open it you find very little in it, but there's a lot of air in it. Why is that air inside that packet? Why it has to be? Same, they are giving in air and they are keeping it airtight to prevent oxidation of that oil. It is fried in oil, right? Imagine you don't give that space at all. The oil which is there inside will easily get oxidized, okay, and it will easily get spoiled. You will get the smell, you will know very well. You buy a standard chips packet like Lay's will have a different smell, and you buy the same thing in a normal shop, and if the person hasn't sold it for many days, it's quite expired, or it has taken 1 week for it to sell, you will easily find the difference. The chips bought in the normal shop and the Lay's will have lot of difference. Even after years you buy, Lay's will remain as it is because the oil has no chance to get oxidized there. But the one which does not have an airtight container there, it will easily get oxidized, and therefore that smell arises. Therefore the correct answer to this question is option D. Okay, now let's move to the next question here. So look at this question here. You have a picture, you have a formula. In fact, given below is a diagrammatic representation of one of the categories of a smaller molecular weight organic compound in the living tissues. Identify the category as so you need to just fill one blank. That is, you have X here, right? Where is X? It is here. You have to fill what is that. Okay, now a diagrammatic representation is given in front of you and you need to analyze what this X could be. A combination of nitrogen base with pentose sugar is called a nucleoside. We all know that, right? A combination of nitrogen base plus a pentose sugar. Penta means 5, right? Pentose sugar is called as a nucleoside. Now the nitrogen base combines with a sugar molecule as it is 1 carbon atom in the glycosidic bond. So it will be easy if you can read this, then only you will understand. More than telling, you have to just see and read. The nitrogen base combines with a sugar molecule at its 1 carbon atom in the glycosidic bond, which is carbon. So you have carbon here, right? By one of its nitrogen atom, usually 1 in the pyrimidines and 9 in the purines. So they're talking about the bases here. Depending upon the type of pentose sugar, nucleosides are differentiated into ribonucleosides and deoxy. We always know that depending upon the base, we classify them into RNA and DNA, because there's one base which will not be same in both, in DNA and RNA. So that is what they are talking here. Therefore, the correct answer would be nucleoside. First thing, we've got a hint, it is nucleoside. So which compound or which thing actually tells you will differ from DNA and RNA? If you see, it will be uracil, right? So the correct answer is option D. I told you very clearly, DNA and RNA will not have the same basis. One thing will be specific for each, DNA and RNA. Okay, so this is what helps you to identify this is RNA and this is DNA, apart from being a double-stranded and a single-stranded a nucleoside or nucleotide. You're talking about RNA and DNA. Therefore the correct answer to this question is option D. Next question here. Okay, which one of the following structural formulae of two organic compounds is correctly identified along with its related function? So you have A, okay, and you have B. Okay, so looking at this diagram, what can you say? You have A and you have B. Okay, see, they are asking us to identify the related function of it. Okay, the correct answer to this question is lecthin, which is option D. Now what do you mean by lecthin? You have any idea about it? See, you can call it any way around. lectin or lectin, whatever you want. It's a generic term of a it is a generic term to designate any group of yellow brownish fatty acids occurring in the animal or in plant tissues. So I'm talking in general about this. Okay, so this is the answer. This is actually a fatty acid. Where is it found? If you see, it will be predominantly present in animals and plant tissues, which are amphiphilic, that is, they attract both water and fatty acids. Amphiphilic means they have two properties. They will attract water and they can attract to acids, to lipid bases also, fatty substances. Okay. So what is the main function of this particular thing? If you see, it is used for smoothening food textures, emulsifying, homogenizing liquid mixtures, and repelling sticking metals. So here, if you see, mostly if you see, let me give you an example here. When you see chocolates, it's a combination of water and fats. Obviously they have fats in it, right? Now these kind of lectin or lectin are used as a smoothing agent because it is attracted by both water and fat, so it can easily mingle with both and thereby give you that smooth texture. Main property of this is particularly they follow in chocolate or food texture industries, where the appearance plays a vital role. Therefore, the correct answer to this question is option D. Okay, next question here. This question was asked in the year 2000 as well as repeated in the year 2006. Okay. Length of one turn of helix in a beta form DNA is approximately dash. So this is just calculation that you need to know. So to your right side you have the formula. Length of one turn of DNA helix is actually or usually 34 Armstrong. Okay, so here they are asking us length of one turn of a helix in a beta form of DNA is approximately dash. We all know that 1 Armstrong is equal to 10 to the power of minus 1 nanometer. So 34 minus will, when 34 comes this side, it will become minus, right? So 34 will become a negative charge, which is equal to 10 to the power of minus 1, which is into 34, which gives you 34 into 1. 34 is, we have taken it in the common manner now. 34 into 1 divided by 10 will give you 3.4. Therefore, the correct answer to this question is option A, 3.4. So what will be the length of beta form will be 3.4 nanometer. Okay, I think it's a simple max that you need to understand. When you take 34 as a common category, it will come only once, right? That is what they have mentioned here. So you don't have to get confused here. All you need to remember in this question is 1 Armstrong is equal to 10 to the power of minus 1 nanometer. Only if you know that value, you can do the calculation, right? Next question. This question was asked in the year 2005. Which one of the following hydrolysis internal phosphodiester bond in a polynucleotide chain? Okay. Now what do you mean by phosphodiester? 2. Right, di means 2. Endonuclease. Which one of the following hydrolyzes internal phosphodiester bonds in a polynucleotide chain? Is it lipase, protease, endonuclease, or exonuclease? Lipase it cannot be lipase because it is associated with the lipids. Protease, it cannot be protease because it is associated with proteins. Right. Endonuclease, endo means inside and exo means outside. Right, so here they are asking us internal. So it's very clear it should be endonuclease. So when you get these kind of questions, always remember to look for a keyword. A hint will be there in the question already. If you read the question clearly, I'm sure you'll be able to answer easily. Now this question, if you see, has given you a keyword of internal and you're given 4 options there. It obviously cannot be lipase because it will hydrolyze it will only work towards lipids. Protease towards protein. Now out of the 2 options, endo and exo, endo is internal and exo means outside. So here they have clearly mentioned it is internal. So the correct answer is endonuclease. Endonuclease would hydrolyze the internal phosphodiester bonds in a polynucleotide chain. When I say polynucleotide chain, they are resembling to a DNA. while exonuclease will hydrolyze the terminal that is present in the outside or in the end. The ones which are outside or which is present in the terminal ends will be hydrolyzed with the help of exonuclease. Therefore, the correct answer to this question is option C, endonuclease. Okay. So with this we complete the biomolecules part 2, which was easy for us. I hope all of you understood whatever I said or explained. Hope to meet you all in the next part of the same chapter. Thank you.